This
Right.
conference will now be recorded.
Okay, perfect. So hello, everyone. My name is Emily Noakes, and I am SECOORA's communication specialist. And today we are going to be hearing from Dr. Jennifer Loch about collaborative fisheries with a smile, size matters, innovative length estimates. So SECOORA is the Southeast Coastal Ocean Observing Regional Association, which is one of 11 regional coastal observing systems that make up the NOAA-led U.S. Integrated Ocean Observing System, also called IOOS. We serve North Carolina, South Carolina, Georgia, and Florida, and we strive to collect data and create tools that support human populations, coastal economies, and a healthy, sustainable environment. Our mission is to observe, understand, and increase awareness of our coastal ocean, supporting community, economic, and ecosystem resilience through strong partnerships. And here are some examples of some different assets that we have and community projects. As a reminder, we do have all of our past webinars archived on our website. You can find it under the News tab. It's called Webinars, and there's the URL if you're interested. You can find all of our archived webinars all the way back to 2017, and it will take you to our YouTube channel if you click on the title of each webinar, and you can watch to your heart's content. We also have summaries of each webinar that we have done in addition to the speaker and the date that the webinar occurred if you wanted to just get a quick recap instead of having to watch the entire thing. So with that being said, I would like to introduce our speaker for today. Dr. Jennifer Loch is a research associate at Reef Environmental Education Foundation, which is a nonprofit, citizen science-based marine database organization. She works directly with Reef's Conservation Science Program to coordinate the SMILE, Size Matters, Innovative Length Estimates project, an initiative to incorporate fish length data into the Volunteer Fish Survey project and supplement fishery stock assessment data using camera technologies. Her work is motivated by advancing marine conservation of fishes for sustainable populations, thus healthier ecosystems. She enjoys engaging with the public, anglers, and citizen scientists to better understand their knowledge of marine resources to ultimately further conservation through collaboration. Dr. Loch holds a Bachelor of Science in Biology from Florida State University, a Master's of Science in Marine Biology through the 3Cs program at Northeastern University, and a PhD from the University of Central Florida. So with that, I will make Dr. Loch the presenter, and you can take us away with your presentation.
Great. Thank you, Emily. And thank you to SECOORA for hosting us for this webinar, and to all of you for taking time out of your day to join us to learn more about our research project. As Emily said, I'm Jen Loch. I'm a research associate at Reef, and I'm presenting on our SMILE project, which stands for Size Matters Innovative Length Estimates. But this is a highly collaborative project, so I want to highlight our partners for this project that are all on this slide. We are funded by a NOAA CRCP grant with a lot of support from the South Atlantic Fishery Management Council, as well as Scripps Institute of Oceanography, as well as UC San Diego Engineers for Exploration program, and our data management partners with SECOORA and Axiom/Tetra Tech. So first, I just wanted to speak a little bit about our organization in case people aren't familiar with it. But I work for Reef, which stands for Reef Environmental Education Foundation, and we're a nonprofit organization that supports a global marine sightings database and related education to advance marine conservation, which is supported by a global network of 85,000 members. So we have a nice infographic here highlighting some of our incredible numbers. But the way that this primarily works is with the most well-known aspect of Reef, which is the Volunteer Fish Survey project, where we train divers in fish ID and relative abundance to conduct fish surveys all around the world during their regular recreational dives. So we have about 17,000 active surveyors within our membership, which have supported over 300,000 fish surveys over the 30 plus years that the program has been in place. This has contributed to over 130 scientific publications. So not only is this an incredible source of data collected by citizen scientists, but it is publicly accessible and accessed as well by scientists. So the way that it basically works is our divers are trained in fish ID and with whatever region that they'll be diving in, as well as metrics of relative abundance. So there's basically 4 bins for that. It's single individuals, few individuals, many, or abundant. And then we also test our surveyors for their knowledge, whether they be considered novice or expert surveyors. So they take tests in their fish ID knowledge, and then it's also related to how many surveys they've supplied over time. So this has been a really incredible resource, as I mentioned. The proof is in the numbers, but still recreational divers are an underutilized source of data collection. But because of all of this time spent underwater, because they're kind of the eyes of the scientists when we are so resource limited in other ways, they can be a valuable source to help fill data gaps in fisheries. And some of that's been demonstrated with a recent publication from last year where we look at the reef data set and compare it to the existing professional science data set with NOAA. This is the Reef Visual Census, or RVC method, which I'll actually talk a little bit about in a second. But they're doing visual census as well, but in a specific area and length of time that they're doing their fish surveys, whereas ours are roving divers throughout their regular dive. So on these different panels, we have reef in red, the reef data in red, and in blue is that RVC data. On the x-axis is time, and on the y-axis is relative abundance. So you'll see that there's a lot of agreement between the two data sets, but we do see variation among taxonomic groups. So if you were to go look at this whole paper in the supplementary material, there's tons of figures on tons of species. So this is just highlighting a few. But what some of the trends are that we see is that there's some variation in that the reef data set is really good at getting a lot of cryptic species, whereas RVC, they're getting kind of whatever fish passes in that area at that time and tends to be a little bit more of the large-bodied individuals. But there is still the highest agreement along the large-bodied solitary species like the black grouper that you see on this picture, on this figure rather. And so those tend to be the species that are of ecological and economic importance to us, do have the highest agreement between these two different data sets. So that's great news, but they all can be really good at different things as well. So we also are kind of trying to add something else to our reef data set, which is looking at size data. And why that is because fishery stock assessments incorporate size data for various purposes. One, it can tell us about population growth or decline. It can tell us about reproductive potential, as well as biomass. Some of those things I'll get into in a minute as well. But the way that we typically obtain length data is through direct harvest, such as through the commercial fishing industry or through directly handling a fish. So usually with the fishing industry, we're getting fish sizes as fish are brought in, which is from a very limited species because it's whatever they're targeting, as well as limited sizes because it's based on their target, as well as their gear methodology. Even professional scientists like me measuring this snook with a pathetically small ruler. I clearly didn't expect a big fish that day. But scientists, when they're handling fish, or recreational fishermen, when they're handling fish, we can get data from those methods as well. And even when the fish are released, this induces a lot of stress and can sometimes have post-handling mortality as well. So we're looking at some other methods to do this without handling the fish that's accurate and expands the distribution of sizes and species because we're not limited to those restrictions of gear and fishing restrictions, for example. So there are some existing in situ methods that are pretty right now limited to scientists. One of those is the RVC, or the Reef Visual Census method that I mentioned earlier, where NOAA divers are really highly trained in being able to identify species from a distance, as well as estimating length with the T-bar that you see there, which is basically a ruler at a set distance. They get their refreshed training every year and testing to ensure that they have the knowledge and the skills to be able to do that. So a lot of resources go into being able to train for that. And then we have camera technology as well. So existing camera technology is something like the stereo video camera that you see in this picture. It's a dual GoPro system, which basically works like our eyes to triangulate how far away the fish is and how big it is relative to the distance. And it uses software on the back end to calculate the size of the fish, which is a pretty accurate method, but it's a little bit bulky and it's pretty pricey. So not very conducive to regular everyday divers. A similar method to that is with laser calipers, where you have your calipers, your lasers rather, calibrated a specific distance apart, and you can estimate the length of that fish in post-processing. So why do we care about fish length? Anybody in the audience that works with fishes is probably very well versed in this, but for those that don't work with fish very much, as I touched on before, fishery stock assessments incorporate size data to really indicate what's going on within the population. So this is an example of some of the data I'll be showing, but something that is reflecting a population where you have a length frequency distribution. So you can see the distribution of lengths within that population in that moment in time. So you would have length on the x-axis and the frequency, how many of individuals are at those different lengths on the y-axis. But it can also reflect your age structure of a population. So fishes that have been researched previously, they'll associate their lengths with their age by they're harvested at that point. You can look at their ear bones or the otoliths and age those fishes and compare it to the length that they're at. So fishes that have been studied previously would have these length age curves associated with them, like you see in these panels, where you have length on the y-axis and age on the x. And so you can kind of like back calculate how old something is based on its length, if it's a known species that's been studied. Obviously, we don't always know exactly what that fish's birth date is or something, but it gives you an estimate of how old it is. So length is a really great implication of age without having to harvest that particular fish. Length is also indicative of reproductive potential, because typically the larger the fish, the older the fish, and the more fecund or higher reproductive potential that that individual has. So it can be also a good reflection of the longevity of that population moving forward. And then similar to age structure, the length is related to biomass as well. So getting just something as simple as how big is that fish can tell us a lot of information about its biology, but also about the population itself. All of these data about their lengths and about their population and what it's doing right now, where it's been before, where we can project it to be going, helps set fisheries regulations. And so that's what sets for both the recreational and commercial fishery is how big the fish that can be kept and harvested. And even those data feed back into this whole process, and you can go through it again. You make adjustments for those fishes over time if they are changing, and it might modify those regulations moving forward. So that leads us to the SMILE project, which stands again for Size Matters Innovative Length Estimates. And this is where we're trying to kind of piece these two things together with Reef, and then to help improve data for our fisheries management and assessments. So SMILE is a citizen science initiative to collect fish length measurements using a low-cost in situ tool. So the goal is to complement the existing reef fish surveys, because now can we get length data on top of relative abundance and diversity, and hopefully being able to improve future stock assessments for data limited species, and especially again to expand the distribution of those sizes so we can maybe have a more accurate depiction of that population and ultimately inform management and conservation efforts of those particular species. So why participate? And this is particularly a message for our citizen scientists, but there might be different reasons that people participate, and we're trying to get into that I'll touch on later. But the way that we see it is you're helping scientists and fisheries managers fill important data gaps. You can get involved with new science. Just like any tech, this is a constantly evolving project. As we get better at it, as we can reduce the cost of it, as we can reduce the time to process data, things like that. So citizen scientists can get involved like at the cutting edge moment of this new technology to contribute to fisheries science. It's also fun and easy. As I'll talk about, these cameras have lasers on them that help us indicate how far away the fish is to estimate its length. So what can be more fun than playing laser tag with fish underwater? It's also pretty easy to use. So it's very little training that's needed to gather this important data. And then lastly, if you're not great at fish ID, like a lot of our reef members are, they're very well versed in fish ID. But if you're new to our organization, learning 300 species in the tropical Western Atlantic may be a little bit daunting to some people, but you may still want to contribute data and enter the SMILE project. We only need you to learn 12 species and have a cool camera with a laser to be able to contribute important data for us. This project is highly collaborative, like I mentioned, but we also, in addition to these partners on my first slide, we have a stakeholder panel that advises us on methods, on and data usage, so how the data is going to be used from here. What other things do we need to collect when we're using the SMILE camera, as well as different site and species selection as we've been in the pilot testing phase of this project, which is what we're in right now. So this is highlighted where our current data has been mostly collected is within Alligator Reef in the Florida Keys. We selected this area because we dove right into the reef database for this to see where most of our target species would be within the Florida Keys, and particularly wanting to look at these sanctuary preservation areas. So that's the other reason that we selected it. It's considered a SPA, or a sanctuary preservation area, and that red polygon on the map shows its current location of that SPA. And it's also being proposed to expand that SPA out to the 90-foot depth corridor. So we've been able to do some cool ecological analyses as we process these data to look at different size distributions within and outside of a SPA, and also in an area where we know we'll have these target species to best test this tool. So with that, I'll show you our curated list of target species for the SMILE project. So the goal for these species is to identify those that are of ecological and economic importance, and may have some different data needs based on our partners with the Fishery Management Council, for example, or state partners with FWC. So we really focus on the typical species for that, like grouper and snapper and hogfish, for those economically as well as ecologically important species. And then we have fishes to be more of an ecological indicator in the Florida Keys, especially because of their herbivore or corallivore status. So we have black grouper, Nassau grouper, red grouper, and Goliath grouper. We ask people, basically, if you're seeing any large grouper, take a picture of it, but these would be the 4 likely candidates that people would encounter in the Florida Keys. Then we have hogfish, followed by mutton snapper. We've just added yellowtail snapper, so they're not necessarily data limited per se, but they've had a recent fishery assessment, so they can be used as a source of validation for these data, and a point of comparison with an existing data set on yellowtail, and kind of seeing how these two things can work together. And then we have gray or mangrove snapper. And then lastly, the 4 species of parrotfish. So there's midnight rainbow, blue, and then the 3 phases of stoplight parrotfish, which are also probably the most common species. So we can ensure that during these pilot testing, we always have somebody out there as well that can be photographed and used to process our data. So these are also pretty easy to identify species. Again, if people are learning them initially, and if people are surveying, they can just kind of identify it on their slate and do both a reef fish survey and take pictures of them at the same time. So the tool that we do this with, our SMILE camera, is called the Fish Sense Light camera. So I'll abbreviate that as FSL later on. This is an Olympus TG6 camera. So this is a commonly used camera with recreational divers, and it's also a really easy to use camera. So if you haven't used one before, this is a good point and shoot camera that's quick and easy to learn. We put it in its typical underwater housing, and then we have slight modifications to that. So we have a detachable wide angle lens, which is currently used to reduce distortion on the edges of the image. We have a cold shoe mount that houses a single laser. So the laser is in a fixed position, and this camera system is calibrated between the camera, the housing, and the lens, and then that known position of the laser. You can change the laser in and out, but the fixed position of it is standard. When people are using the camera, it does need to have specific camera settings, and I don't need to get into the details of all this, but basically when somebody's getting trained, they are going to get up to speed about what the camera settings are. But it's still easier than this, because basically if you have the camera set to P or program setting, everything else should be correct. But we shoot without the flash with the ISO at 1600. We shoot in burst mode so that we can ensure that we are getting multiple images of an individual, so we can build the best length estimation. because they are living, breathing, moving things. And so you're not always going to have the best angle with just one singular shot. We keep the accessory and face priorities off. We shoot in autofocus, things like that. And then the most important aspect for the measurement possibly is that we have an output format as the raw image file. So every image has an associated raw file that goes with it. So that always has to be in place when using the FishSense Lite cameras for this project. So for our data collection, we've been doing this in the Florida Keys, as I mentioned, but we've also kind of done things in Key Largo. We've taken it on different reef survey trips. So these cameras have gotten around in other areas as well. But when we're doing our pilot testing dives, the procedure that we do is we have citizen scientist divers use the FishSense Lite camera with their prior training. And then they try to get the laser on the lateral side of these 12 target species. So it doesn't have to be perfect, which it is with this parrotfish, but they can deal with a little wiggle room on where that laser is placed on the fish and like slight angles with the fish as well. But that's the goal, is to try and get it as broadside lateral on the fish body as possible and central if you can. We compare these with the existing methodology, the existing camera technology, the stereo video cameras, which are used by staff to compare with those FishSense Lite cameras. So we'll do this with paired dives, which is some of the data I'm going to show you today. Basically, a diver with the stereo video camera is paired up with someone with a smile camera, and we try to get the image of the same fish to measure that same individual between the two camera systems. We do this with roving stereo video, so we just go around the dive just like anybody with a smile camera would. And then we do it with a known distance as well. So we also have 30-meter belt transects with stereo videos to compare any densities and things like that with fishes and sizes in a known area versus roving. So the basic procedure for using these cameras is that initially you need some brief online training that I mentioned, and then we have dockside instruction. So before somebody goes to use the camera, we just kind of show them how to turn on the camera, how to turn on the laser, and then how to work with the lens. Because as you're diving down, we do ask divers to flood the lens or burp the lens. Basically, they loosen it and then tighten it again just to flood water between the housing and the lens so that you don't have air bubbles. And then prior to them going down, checking the camera settings or having a staff member check the camera settings. But as we move this project forward, those are things that citizen scientists would be doing. So once they've checked their camera settings, they're going down, they've flooded the lens, they've turned on the laser, the camera's on. The initial procedure is that they take photos of a preset dive calibration slate, and they would repeat that process at the end. So they take photos with their dive buddy holding a dive slate, and then after that, they just go and perform their dive like usual. We've just asked them to dive as they would dive, and they're gonna, if they encounter any of these target species, they'll aim the laser of that camera on the central lateral side of the fish, hold in burst mode, and capture the images of the fish with the laser on its side. People that are trained in fish surveys with Reef can also perform a fish survey simultaneously. So we have divers do that as well. So we can get feedback on how these methods can work together. And then after the dive, they will take pictures of that dive slate once again. And then once we're topside, we've asked them to record some metadata, like where they were diving, when, what time, any camera issues, as well as indications of the dive conditions like visibility and current, things like that. And then all those images are downloaded and they're supplied to the engineering team at UCSD, where an AI workflow can determine the length in post-processing. So the citizen scientists don't even need to be measuring the fish themselves. It's literally just a matter of them taking pictures of the fish and supplying that to us. So it's a easy to use, easy to learn, high quality source of data. So the way that it works, this is kind of the common question that I get from colleagues in our field, is like, how are we doing this with a single laser? So the way that it basically works is it works with depth of field. So if you imagine things that are farther away appear smaller, things that are closer up appear larger. But we need to know what the actual size of that is. So we use depth of field to our advantage. So if you have an image like this on the reef, you imagine that there's coordinates within that image, things that come from the raw data file. So they can actually tell the position of that laser within this image. So the fish that's highlighted there with a red laser, oh, sorry, these little numbers got shifted on the bottom, but that has a known XYZ coordinate. that if you were to get closer to that fish, the laser position now shifts, and the position of that laser would have a different XYZ coordinates. This is where it was in the past in that open circle when the fish was farther away. As you move closer to that individual, the coordinates of that laser change. So that helps us know how far away the subject is from the camera, because that is influential in estimating its size accurately. So the next step is identifying the head and the tail of that individual fish. So we can identify the laser, it's red on this individual, locate its head and tail. And then with the raw image file, they can tell the number of pixels that fill in the distance between the head and the tail, and the known distance of the laser from the camera with that coordinates can tell us the length of the fish. So it's able to compute the fish length. We compare this with stereo video data processing. As I mentioned before, that's the system with the dual GoPros that have, it's got it in both cameras that are synced. And then we use the CGIS event measure software to compute the length of those fish, which is still a manual process as well. We know there's other systems out there like Vimeo that can assist with these things, but for what our specific needs are for this, we have our developed AI workflow for that. So some of the preliminary results here, this is work with paired tests. So this is like, oh, there's a lot on this figure, I realize that, but these are all individual fish. So you don't need to worry about the stuff on the bottom, but that's my proof. They're all different individuals found on a reef that have been matched up with a stereo video camera and a fish sense light camera. So side by side, confirmed individual. They're not going to be the exact same angle, so that'll lead to some variation a little bit between the two systems. But on the y-axis, you have mean fork length, so we measure straight through the middle of the fish. The fish sense light are in tan, and the stereo video is in blue. So there's some variability, especially among individuals, but there is a lot of agreement between the two camera systems. So there's no statistical difference between them, although you have some little outliers here and there. But the stereo video system does tend to measure a little bit larger. And we see that with some of our length frequency distributions as well. So this figure is different species, but in the top panel in A is all of those fishes from the previous slide, but now displayed with their length frequency distribution. So the x-axis here is fork length and the frequency is on the y. Same colors for the camera systems. The tan is the fish sense light, so the new smile camera technology. The blue is the stereo video, that existing GoPro technology. So they are quite similar in capturing fish length. The means of these two camera systems for each panel is shown in those vertical dotted bars. So there's no statistical difference between the means, even though the stereo video, again, does measure a little bit larger. That is more dramatic in these following panels for midnight parrotfish, gray snapper, and hogfish, where we have lower sample size basically, which can lead to larger differences between them, but they were still not statistically different from each other. And those means get closer together with the higher sample size, which I guess is to be expected. So with stoplight parrotfish, which I mentioned is probably our most commonly encountered species, and for blue parrotfish on that day, much closer means. But again, none of them are statistically different, and the stereo video just measures a little bit larger. So you may ask which system is right, and we also ask that same question. So when they're developing these cameras at UCSD, they do pool tests with their known objects, but we'll do them as well with both camera systems. So we've done them in the pool and we've done them in the ocean. So we're continuing to process these data because this is the same color assignments for the camera systems, but we just have one fish sense light camera on here. We are actually up to 11 camera systems for the fish sense light for the SMILE project. And then I have it compared with two different stereo video systems here. So this figure is camera type on the x-axis. The y is mean length of our little model fish species. So we take down fish models in the pool. I love taking them on the reef just to confuse real fish, but that gives us the same conditions that we would be measuring stuff with our live fish. And then the horizontal dotted line here is the known length of that fish model. So both systems are pretty darn close to measuring that, but we see stereo video is just slightly larger. So we'll continue teasing that out. Maybe it's something too with species variation between the different species that we'll look at as well, with what system might be larger, smaller, has more variation, that kind of thing. So those are just some of the example data that we have for our project so far. Luckily, we've had a lot of success with this project. One of our, I think really our only main problem is the lasers. The laser performance is fine, but the laser longevity is not. So we have a lot of laser flooding that happens with not a lot of pressure that really gets put on them. So they tend to flood with minimal use. So we've been through lots of different laser types, and now we're just developing our own with our partners at Backscatter. So we're developing one that does have a dual parallel option, or you can still use it as a single laser, maybe for smaller individuals, or divers can just use it as a pointer. But if we have the dual laser system on, that would actually eliminate the need for the calibration slate at the beginning of a dive. Those calibration slates just tell the engineers if it's drifted at all during the dive, so this can potentially eliminate that. We also want our lasers to have a rechargeable battery, not being able to be like twisted open and closed like a push button on, so we can eliminate some of those sources of flooding. And we've also developed a more robust mount. So we had been using 3D plastic mounts. So that's the other challenge, is those get pretty brittle over time, just being in salt water. And so they break, and it leads to lots of change out of the mounts, which can be done on the camera. Again, we just know that it's in its fixed position. It doesn't have to keep that exact laser mount because they're all made identical. The other component to this project is the citizen science aspect. So I've been talking more about the fisheries ecology right now, but to me, I see it as two things. It's fish ecology, fisheries management, and it's citizen science. So we can't do this project without them. And so far we've had 46, actually this number's changed now. They just did a workshop this weekend. So I need to update my number, but we've had probably around 50 plus different volunteers on the project. A lot of them are actually repeat divers. So they like playing laser tag with fish underwater, so they come out with us again. But we've done over 300 total test dives in the Keys at this point, and the cameras have been on 7 different reef survey trips. So Reef hosts different trips around the world that people can get involved with and learn fish identification. We've sent those on some of the local tropical Western Atlantic trips, and it's about to go on another one. So we try to get people more exposure with the cameras and also using them in different areas. We are starting to build up, especially now that we have 11 cameras, local dive operator partnerships. So we've already got one dive operation doing this with us. We've dove with so many different groups that we have great partnerships, thankfully, with in the Keys. But we're trying to get that out with their clientele. So having a streamlined process where they have a quick webinar that they can view dockside or on the way out to the site. They get a little bit of training on the fish ID and a card that they can see the different species on. And then just shown how to operate the cameras by the trained staff at that dive operator. So we can get the cameras out to more people and kind of spread the word about this source of data collection and about reef and in general. So, and then the other thing that we have for our citizen science aspect is a formal survey that we've developed with colleagues at Colorado State University, because as we started doing this project, we were wondering like how to engage with different groups and what interests people, how can we get people to kind of collect more data. And Reef has never done a formal survey like this before. So we've been developing this, and it's out into the world now. I'll ask you to take it even here in a minute, but with access to it with a QR code. But it's to examine motivators and barriers to participating in this project, but also conservation technology more broadly, maybe even marine monitoring more broadly, and also specifically looking at camera performance and use. So as I said, this is our first ever formal survey. This is a QR code that'll direct you to that source. It's like 10 minutes or less, really, we say 10 to 15 minutes, but I don't even think it takes that long to take the survey. Again, to examine motivators and barriers for participation in this project. So we ask things like, what would be your level of interest in using the camera for this project? But more broadly, if you participate in marine species monitoring, how much of an impact do you believe your contribution would have on fisheries management? We give you options and even an area to fill in, like what would motivate you to get involved in projects like this? Is it just to get involved with the community? Is it just to do something new? Do you like tech? Or is it to contribute to the greater good by providing these data? We also have specific things like what type of training would you prefer? And then we have a post survey for divers that have done this and then used the camera to give us direct feedback on using the camera and getting involved with the project. So we would say, For example, if you surveyed, you would say yes, and then it would lead you to a question that was, how easy was it to perform both a reef survey and the camera for the SMILE project? So we know moving forward how well we can implement this into the volunteer fish survey project that's in existence at Reef. So lots of different informative questions that we will be analyzing at the end of the fall and preparing for a peer-reviewed manuscript. These things will also help us understand the target audience, because there's questions about demographics. So we can kind of best know how to cater this project to get more people involved, and again, just get more data for areas that need it. So we put our citizen scientists to work. So not only are they diving with us, as I've mentioned, not only are they taking surveys for us, but we have more recently gotten them involved in the post-processing as well. And they love it because they've wanted to see kind of how the images look when they're done with the project, which they don't always have access to in this pilot phase, just because data are getting uploaded by staff, and getting more involved in the process. And if you're not a diver, you can help contribute to this project by doing AI annotation for us. So volunteers perform really brief AI, they do training for training the AI. It's a really brief online training, and they have direct access to us for any questions. They have online instructions, and then access through this program called Label Studio. So it's all like an open source program, but we directly give the link to them, so we can have some control over the security of that. But they basically do three main components right now. It's identifying the laser location, so that would be on this right side panel. Identifying head and tail, that I'll show you in a second, and identifying fish species, so that we can build the AI to start doing that more automatically for us. We'll also start having them do some like body contouring. They call it like painting of the fish with your cursor, to help the AI start finding fish better as well. So the way, okay, it's gonna work. So the way that the Label Studio works is, for the head and tail annotation, you would zoom in on the image. This has already been marked for its laser. You use a hotkey to locate the snout of the fish, and a separate hotkey for the tail. Once you're happy with those locations, you would hit submit on the image, and it'll bring up another fish for you. It's not like set in stone. If somebody makes a mistake, they can delete that mark and correct it. We go through and check randomly some images to make sure that people are doing them properly. But we have tens of thousands of images that are being annotated, that have been really moved forward because of our volunteer base that's been doing this for us. So it's another great way to get them involved. If it's something that you want to do, you can reach out to me. I'll have our email address here at the end. It's kind of a great background thing if you're on a meeting or a webinar like this and you just want to keep your mind a little bit active. You can mark fish doing annotation. You can kind of do it while the TV or a podcast is on. So it's been a great way for people to get involved in the process. Again, when you're not wet, you're dry, can help us out. So in summary, the camera systems are comparable to each other, meaning the Fish Sense Light for the SMILE project and the stereo video existing technology are comparable to each other to generate lengths, even though stereo video tends to measure a bit larger, and we'll continue to work that out with our analyses. We've had some troubleshooting for the hardware and software challenges, mostly for hardware in terms of the laser longevity and robustness and the mount and things like that we've been working through. And then we've had a really positive citizen science response. So even though we're getting more formal with that with our surveys, all the feedback that we had gotten in the last 2 years of diving with citizen scientists with these cameras has been overwhelmingly positive. And that is proof to me when they keep wanting to come out and use the camera, and they're really excited about it. Their learning curve is pretty small too. I think it takes like 1 dive, maybe 2, for somebody to get comfortable with it. And even then, they're not going to collect like bad data. It's just you might miss fish because you're trying to track the laser on them, that kind of thing. So it's a fun little challenge, but that actually has a pretty small learning curve. And then we will have these data publicly available, similar to how Reef's volunteer fish survey project is. We would have metadata available specifically to our end data users as well. You can get more of the specific data as needed. And we look forward to continuing to build that out and have platform that divers can upload the images, that kind of thing, as we continue to scale the project up with our dive operator partners and others that are using the cameras on a more regular basis. So with that, I'm going to end on our infographic that's been developed with SECOORA and the South Atlantic Fisheries Management Council. As I like to joke, everything I just said in this talk is kind of truncated here in this nice infographic. So thank you for your time, but you get it kind of nice and summarized here. Our QR code can direct you to our website where the survey also is, and then our email address is there at the bottom where you can get in contact with us if you want to get involved with the project in any of the various ways we talked about here. And with that, I'm happy to answer any questions about the project. And again, I thank you for your time and listening today.
Thank you, Jen. Like Dr. Loch said, we are going to open the floor to any questions. You're welcome to put your questions in the chat, or if you feel comfortable, you can raise your hand and turn your camera on and say hi and ask your question on your own. I'm going to kick it off with my own question. So I'm wondering if a project like this could be transferable to another location in, maybe another part of the world or another part of the country. Do you think that something like this could be useful for other big fisheries zones?
Absolutely. Yeah, absolutely. So we've been pilot testing it in this region. But not only do we have a lot of camera systems, the other goal with developing our own laser and the mount is to get this as affordable as we can so that divers can eventually purchase this attachment themselves and be able to take photos with their own cameras. So that's certainly down the road for us as we're developing the tech a little bit better, but that is an end goal for us to be able to get it in the hands of more people all over the world. And that's also a way for us to do that with the reef survey trips too, is to kind of capture some of these regions and get feedback that way too.
Okay, and kind of a follow-up to that question, do you have to have the super clear water and the shallow reefs to be able to use these lasers, or could you do it in deeper water with less light, or in water that is maybe more turbid?
Yeah. So you can do it in more turbid water. They actually test them in San Diego. So they're doing their initial tests in water that's not as clear as the Caymans in that image. The better the visibility, the better you're going to be able to mark the head and tail of those fish. You're going to be able to see the laser a little bit better. Lower water visibility, or more turbid water, the fish tend to see the laser beam a little bit more, so they can be a little bit trickier to track down. But it's not outside the realm of possibility for sure. Like and we're in the Keys. I mean, we are not dealing with great visibility all the time. So you may just be getting individuals that are a little bit closer to you. It's like a maximum of 5 meters. 3 meters is probably the sweet spot for getting your images. So I should have mentioned that. But yeah, you probably just need to get closer to your fish, or you're just not going to have as many. But you can certainly use it in different conditions.
Okay.
I don't think nighttime. Yeah.
Oh yeah, that would be interesting.
Yeah. Because we don't shoot with flash. It creates literal backscatter. So
yeah. Well, does anybody else have any questions? You can just raise your hand, or you can just turn your mic on and chime in. We'd love to hear from you. Okay, Judd Curtis, if you'd like to turn your cam or your mic on, you're welcome to ask your question.
Hi, Jen. Thanks for the great presentation. You might have mentioned this during your presentation in April to the SSC. And I think one of you're collecting a bunch of environmental parameters, and I think depth was one of the other kind of data inputs that you took into there. Can you describe, are you guys seeing differences in depth in any of the species so far based on, the comparisons with the CV, or just the observations from the divers, and kind of what ranges you're operating in?
So when you say differences, do you mean in people's experience with the camera or in the fish sizes?
I'd say in fish sizes. Yeah. So I'm thinking, for again going in the stock assessment inputs, we see from harvest oftentimes differences in size the deeper you get, like larger fish in deeper waters. I'm wondering if you're seeing similar patterns with the laser observations.
Yeah. So I don't have enough data completed yet from the Fish Sense Lite, the Smile cameras. For that, I have some stereo video data that I've been playing with recently, because we're looking at it within and out of the spa too. So certainly we're going to need to look at those variables in our modeling to see if it could be an impact of the protection status versus depth, because some of the species I was a little surprised on were a little larger outside the spa, but then again, they're at depth. So something like Goliath grouper was certainly larger at depth, and they're not heavily harvested or anything. So the spa may not be as much of an issue for them, but that's something we're going to look at while we've been doing this as pilot testing for comparing the two camera systems. We've been curious about the depth contour as well as the protection status. So I definitely think depth will be a strong indicator of length as well. But something like hogfish are really small at depth outside of the spa. They're bigger inside. So hogfish have kind of my expected trend.
Yeah, great. Just something to keep in mind as you're continuing to develop the program. I think that'd be some critical information for stock assessment purposes. So
sure. And we've been talking about that with our data management partners for data visualization to make sure that we include those depth data, and discussing amongst ourselves too about average depth versus max depth. So and if you have any input and opinion on that, you can give that to us as well. Right now we're reporting max depth. So, because we know we can't give everything in a data visualization, but that can be accessible still to the end users that would like that data product. Thank you.
Okay, so the next question is from Chip Collier. He asked, with the citizen science identification, is there any feedback provided to the scientist if there are issues with their identification or length measurements?
Yeah, I don't know that they have any. Some of them become concerned that they're going to give us bad data, but we always tell them that is not a concern for us because you're taking pictures. You're not actually measuring the length. So not so much about the length measurements, but they will sometimes they're taking pictures of extra species, and that's fine because it's all part of the initial data collection process too. So they'll take pictures of schoolmaster snapper, and we're kind of like, when in doubt, if you're not sure, just take a picture of it. It's fine. We'll deal with it later. So we have plenty of species that aren't within the target list. But I think that's really and I guess like issues we've tried to tease out if it's a species thing when they run from the laser or not. They swim away from the laser. But it seems to be a really individual thing. So that's the only challenge. We prefer to work with a green laser these days because it's a stronger color. It does not matter in post-processing what color we're working with as long as you can see the laser. So green is a little bit stronger, but it doesn't influence the analyses otherwise. But the fish can be a little bit more finicky with the green color. So some individual hogfish, for example, bolt before that laser's even on them. Other ones could care less. So it's kind of variable. So those are the kinds of feedback that we get. But something like a black grouper runs no matter what. Something like a red grouper doesn't care what you do to it. So the fish behavior is always at play, which is very interesting. And I give some of that in my training as well about when we're going through the fish species.
That is really interesting. I almost wonder if that could be a project on its own, is how they react to the different light colors and everything.
It's a master's thesis.
Hey, right?
And then something like yellowtail and grunts, they chase the laser, so then you're trying to get a picture of something else and they're chasing you. So there's just a whole spectrum of reaction to the lasers that is pretty entertaining. But we can still get it. We can still get what we need.
That's super interesting. So one more question is from Theo Yas. He asked if there is a way to correct for foreshortening errors if the fish is at an oblique angle to the camera, or is this more or less accounted for with the burst photo method?
Yeah. So this, in my opinion, is a challenge with both camera systems, whether you're using a stereo video or the fish sense light. And there's pros and cons to both when you're dealing with fish at odd angles or when you're dealing with fish that their tail is curved. So we can't reliably use those data. I think it was Scott Happel that said it 15 degrees is their reliable angle, like up to 15 degrees away is their reliable angle for stereo video. So that's probably what we'll be working with. But the engineers at UCSD have more of the specifics for the fish sense light camera and what their acceptable angle is. But yes, for using the smile cameras, the burst photo method helps us get, the angles that we can work with because it gives you more options. The stereo video kind of does too because it's video, but as I say, you're trying to get it on both a left and right camera, and that's a challenge as well because you might have a good view in your right camera but not your left and so forth. So it's just kind of a little bit tricky sometimes, but I always say to mark it, and then we'll get the angle measurements. At least with stereo video, fish sense light will have to be a little bit more subjective, I guess, with the engineers on the angles that they can handle. And that's where those known fish models come into play too. We've done different angle tests with them as well that we're processing. It's a good question.
All right, so that's about our time. So I just want to say a big thank you to Dr. Loch for presenting on this project today, and a reminder that this has been recorded, and I will get it uploaded to the SECOORA YouTube channel and the SECOORA website within the next day or so. And we will also be sharing on SECOORA's social media. So if you know anybody who really wishes that they could have attended today, you can send them the link that way. So as always, thank you. Yeah, go ahead. Do you want to say a?
Oh no, I was just saying thank you. Sorry, didn't mean to interrupt, but thank you guys again for having me and taking time out of your day.
Of course, yes. Thank you everybody for attending. And as always, feel free to send me an email if you think of any more questions for Dr. Loch or any more questions about the SMILE project, and we will get you connected.
Absolutely.
All right. Well, thank you so much, everybody.
Thank you. Bye.
Bye.